Hello, everyone. This is a new class of LinguaLee and AISC and Michelle Wright, and today we'll speak about biodiversity and how species vary according to their different ecosystems. Let us approach this subject field. Organisms diversity. Okay. In 1831, a naturalist whose name was Darwin was hired in the Beagle HMS. And this trip lasted 5 years. Over there, he discovered a lot of stuff about biology. Among those things, he discovered that species presented a smooth variation. For example, in this image, you can observe that variation in one species of beetle. The wing cases of 14 spot ladybirds, Propylia 14 punctata. It showed variation in coloration patterns between these individuals, and they were collected from the same nettle plant. So this provided Darwin a vast information in order to stay his central pillar that was called natural selection, okay, and evolution posteriorly. So he published all of these findings in his book that was called On the Origin of Species by Means of Natural Selection, and then it was published in 1859. Charles Darwin noted that all species showed a large variation. Each individual was slightly different from others in the population, as you can observe in this figure. And this observation formed a central pillar of his theory of evolution by natural selection. And this was an explanation on how all species on Earth could have been originated. Darwin also observed that humans could select different species on Earth and they could breed different animals. Okay, so they could select for different breeds of animals. For example, the pigeons, and that if this artificial selection could lead to new varieties, then there was no reason why. Given enough time, nature could not select for new varieties and ultimately new species, hence natural selection. Okay, so basically what he understood was that as human beings could select for different breeds of animals, for example, pigeons, and that if this artificial selection could lead to new varieties, then there was no reason why, given enough time, nature could not select for new varieties and ultimately new species, hence natural selection. Okay, so there we're going to have a continuous variation. And also Darwin noted that all species showed variation. And some of these variations could be classified as continuous variation. So they are basically at any value of measurement for specific characteristic. In this case, we're going to see a representation of height. Height, that is one of the representations of continuous variation can be represented in this normal distribution. So many features of humans are controlled by polygenes. Okay, when I say polygenes, I mean several different genes that are coding for specific trait. So these polygenes can include body form, can include weight, can include height, and they are going to be related to continuous variation. So a graph of height of a population of 400 people can be represented in this figure. So it will show a continuous variation between the shortest person, that is going to be 160 centimeters, and the tallest person, that is going to be 184 or 185, sorry. So it will have a mean of 173 centimeters that is represented here. So human height is determined genetically by interaction of the alleles of several genes. So in this case, we're going to have several genes interacting with their different alleles, and they will be coding for a specific trait that will be continuous. Got it? So variation in the height of adult humans, and the results cluster around a mean value and show a normal distribution for the purpose of this graph. So the heights are collected into arbitrary groups of each of height range of 2 centimeters. So the human height as a case of polygenic inheritance. But also we're going to have this continuous variation, okay? So the normal distribution means that when the frequency of measurement classes is plotted against the measurement classes, a symmetrical bell-shaped curve is obtained with values grouped symmetrically around the center value. Another factor that may affect the appearance of the phenotype, which means the physical appearance of an organism and its internal physiology, and produce variation between organisms, is the effect of environmental conditions. So this also can affect the continuous variation. I mean, there is a new subject field that is studying this. Currently, is one of the most emerging sciences or derivative for science that is called the epigenetics. So scientists have discovered that organisms can be directly affected by the environmental factors, and those environmental factors can affect the genetic code. At the same time, it will affect the genotype. Posteriorly, it will affect the phenotype. Okay. So, so here we can exemplify this, what I'm telling you, just by placing like 2 different plants. One of them is going to have a correct nutrition just by taking the correct proportion of ions, of mineral ions, but the other one is not going to take like, the proportion of the adequate percentage of those mineral ions. So one of the plants that will be in the correct environment will have the capacity to grow taller and in a certain way healthier, but the other one is going to be shorter. The same can happen in humans. They can be greatly affected by the levels of nourishment received, particularly as children. So the phenotype of an organism is going to be the product of both the interaction between the genotype and the environmental factors. So there we're going to talk about, this continuous variation. Okay, so there are some factors or traits that cannot be like expressed as continuous. They are kind of answer of yes and no, yes and no, yes and no. One of them can be a blood type, and so it's known as discontinuous variation. Discontinuous variation is a variation that has distinct groups that organisms belong to. They have no intermediate data information, okay? And they don't overlap between the 2 phenotypes. Characteristics that demonstrate discontinuous variation are controlled by a single gene. So remember that in the previous variation that was continuous, we were coding like certain traits just by polygenes, no? But in this case, we're going to talk about single gene, usually with 2 alleles. Remember that an allele is one of the variations of certain gene. For example, I can have a gene that is going to code for the eye color. Okay, so for the eye's color, let us say that. So the expression of that gene when the eye is going to be blue is going to be related to a different type of expression of that gene that will be a different allele to that one that is brown, to that one that is going to be green. Okay, so alleles are a representation or a variation of genes. So in this case, these continuous variations are controlled by a single gene, usually with 2 alleles. So the human blood group system has 3 alleles. So this is an example of this continuous variation. But this is not subject to environmental factors, as in the previous type of variation we're seeing, and is determined solely by the genotype, which is related to the genetic composition of the species. So in this way, species can show both continuous and discontinuous variation. Species can show, for example, a certain blood type, but at the same time, it can show a certain height level. Okay? So the same happens here. So an example of continuous variation is height of humans. So the number of people, it will show us a normal distribution that is a curved bell-shaped graph, and at the same time, we're going to have blood groups here. Okay, so distinguishing between different species. How can one species be distinguished or differentiated from another species that also shows significant variation? So as you can observe here in the example, we're going to have a horse, and they will have analogous structure, sorry. So horse legs will be closely related to these insect legs, but this is an ant's leg, okay? So, but they are going to be different. How can you differentiate it? So the normal distribution previously shown states a characterization where morphology, that is a form of the organisms, can variate. So it means that the organisms vary in form and structure. For example, in this case, we're going to have a fish, and this is a fish sculpin fin, and we're going to have the dolphin flipper. Okay, so they will present analogous structures, but they are going to be different species. Okay, so we have the insect here with its wings, and we can have, for example, a bird, which is a chicken with its wing, but those structures will represent different morphologies. Okay. Also, we'll have to talk about sexual dimorphism. Dimorphism is a characteristic that is presented in males and females when talking about different species. Historically, taxonomists, scientists who define and classify groups of biological organisms based on shared characteristics over many years, have learned to distinguish one species from another. So in this case, for example, the most colorized individuals are going to be closely related to be males. Okay, so in that way, they can attract the attention of females in order to reproduce. So many species show sexual dimorphism, where males appear different from females. In this case, we're going to have like ducks, and male on the left, and the female is going to be on the right. And the same happens with the peacock, which is with its feathers, color feathers, to attract the female that is a peahen at the right side. Okay, so this is another concept that we can approach today. It's important to understand that not all the species are going to present the sexual dimorphism. Some of them are not going to be easily differentiated by us, but we're going to talk about that. Okay, so taxonomy, which is a field, will provide a structured way to name and classify organisms. For example, you can observe that it is as an inverted pyramid, where we're going to classify the organisms as species, genus, family, gender, order, class, phylum, kingdom, and domain. Okay, so they will be characterized and categorized based on physical traits, but advances in molecular biology, in genetics, are going to provide us a vast framework to redefine these and to refine this process. Museums are more than just collections of specimens. They are research hubs where scientists store, analyze, and classify biodiversity. Many now house molecular laboratories dedicated to DNA analysis, and also to rethink that process of classification that once was stated by these scientists, whose name is Carl Linnaeus, who proposed the first classification system, or at least one of the first, because before him we have had a lot of people and naturalists that had classified the organisms, but in a wrong way. Okay, so with DNA profiling, taxonomists can verify existing classifications and make corrections, identify and categorize new species accurately, and they can define clear genetic boundaries between species. So every single year, they can, for example, classify a species in a different clade according to their new findings. And it has happened. You can't believe how many times it has happened. So some species look identical, but genetically speaking, they are going to be distinct. They are going to be called the cryptic species. Others can appear different, but belong to the same species. And DNA profiling ensures classification based on genetic reality, not just the appearance. Okay, so the concept of species is based on the organism's appearance. So we're talking about color, shape, and other distinctive features. And this is what Carl Linnaeus brought about. He brought out to our tables this concept that is called the morphological species concept. Even though the concept was criticized, is still widely used, retaining an important place in biological science because this is quite practical and it has some advantages. For example, the first is that is applicable in sexual dimorphism in extinct species. It does not require a gene flow information. It does not require like a high technology capacity. And at the same time, it's going to be practical, easiest, and fastest. So it has also some other limitations, such as it relies on subjective criteria, and researchers may disagree on which structural features distinguish species. And also, different individuals in a species may appear very different, such as males and females. Remember the previous concept that we approached some slides ago. Okay, and so he proposed a binomial system where he was going to classify species according to their genus and species. So the second word that will appear here is going to be the species. So we're going to have the aptenodized forceri, that is penguin, Panthera tigris, which is the tiger, and Danaus plexippus, which is the butterfly. So the binomial system was proposed by these scientists, and there are currently 1.8 million described species using this system. The science of classification is called taxonomy. It can change every single year according to the new discoveries and advances in science, as I told you. So the word taxonomy comes from taxa. In singular, we're going to say taxon. And this is the general name of or groups or categories with a classification system. The process is basically, first we're going to give an organism an agreed name due to a convention in science, and then we're going to impose a scheme upon the diversity of living things. So Charles Linnaeus, or Carl Linnaeus, a Swedish botanist, physician, and zoologist, developed this method in 1730s, sorry. So the binomial system, meaning two parts, he studied with plants, classifying them by their sexual organs. So each plant had a Latin name, which is the genus, and it was followed by an adjective that is species. So it must be written in italics or underlined. So here I want to illustrate an example, in this case, with this species that is related to us. So Homo neanderthalensis is one of them. but you can observe that it has the same genus, that is going to be Homo antecessor, which is the next one, the Homo sapiens and Homo erectus. They will have different species, but they are going to belong to the same genus. So they will have like a relationship. They will be closely related one to each other, and they will have different evolutionary processes. But after the genetic name is shortened to the first capital letter, why? Because look, this is a scientific convention. This is a common student's mistake. So when you're going to express a scientific name, you start by capitalizing the first letter, but the second word is going to be in lowercase. So the lowercase will indicate the genus, the species, sorry, and the capital letter is going to indicate the genus. So that's how it's done, guys. Okay, so let us approach to the next. Let's talk about the next slide. They will differ in their species name. Okay. So in the scientific literature or literature, Homo sapiens can also be expressed as age period sapiens. Why? Because, okay, according to the scientific convention, we can write a full name at first time, but then as we're going to continue using it, it's not necessary to continue writing the full name. But according to the scientific convention, we can just write the capital letter, the initial of this of the genus, the period, and then species. That is a convention because it will take a lot of time, a lot of space, and it's not that practical. Okay, so let us see the next one. So, System and Nature was a book that was published by Carl Linnaeus. So he studied his famous work here, showed for the first time how life could be classified, proposing the binomial system in the genus. That was a group of similar and closely related species. So double names for organisms in Latin, and with the generic name preceding the specific name. Okay, so here I can illustrate you this with two examples. Panthera leo, that is the famous lion, is going to be described with the scientific name. And then we have the Panthera tigris, which is a tiger. So naming organisms by the binomial system has been highly effective. Okay, so as I told you, there is a common mistake, and the common mistake consists of just by putting the lowercase letter in the initial of the name. For example, Homo sapiens. So instead of writing Homo sapiens with capital letters, students have the tendency to write the H as if it was like a normal word. But when you're writing scientific name, lower cases in the genus name. So this goes with capital letter and this goes with lowercase. And we're gonna use italics. Got it? Okay. So let's see what's next. Okay, so here we have nature of science. Science as a shared endeavor. Why is a standardized universal method for naming species needed? Okay, so as you can see, many organisms will have local names assigned by local populations, but these names often differ in different parts of the world. So they do not allow observers, scientists to be confident about the different researches, and they will, for example, use names such as the magpie. So we're going to have an example like the magpie. it represents entirely different birds commonly seen in Europe, but also commonly seen in Asia and Sri Lankan. Okay, so we're going to have, for example, the European magpie, the Asian magpie, and the Sri Lankan magpie. So all of them are going to be similar, but they will belong to different species. Okay, so for example, we have the pika pika. We have also the Platysmorus leucopterus, and we have the Urosticea ornata. All of them are going to have similarities, but their scientific names are different. So instead of using their local names, scientists have settled down a convention, and they will use the international binomial system so that everyone around the world knows exactly which organism they're talking about. So this will allow scientists to cooperate among themselves to continue developing and applying different researches, and this agreed convention and common terminology for species names used globally helps to prevent ambiguous communication. So there is another concept to determine species, and that is the biological concept. It will focus mainly on the reproduction that can be presented in individuals that belong to the same species. So this concept was proposed by zoologist Ernst Mayr in 1942. And this biological concept will present some limitations because there are certain limitations that can be seen or observed here. It doesn't work with asexual organisms such as prokaryotes or neither for extinct species. And it overemphasizes gene flow and downplays the role of natural selection. So, as natural selection can cause something that we call speciation, many pairs of species that can be morphologically and ecologically distinct to remain distinct, and yet have genes flow between them. So it has some limitations too. But there are other alternative species concepts. We have also the ecological species concept, where each species has a particular role in the environment. So the niche of species is everything about where and how it lives, what it is, how it reproduces its habitat, and so on. But the problem here is that we have not determined all the niches around the species. So it accommodates asexual and sexual species, which is a great advantage. But generally speaking, niches are generally difficult to be identified because they involve the interaction between biotic and abiotic factors. Another advantage that we can note here is that it emphasizes of the role of disruptive natural selection or organisms adapt to different environmental conditions. But okay, as far as you know, we have not determined all the niches, all the niches in all the organisms. Okay, today molecular science has allowed us to identify other species. So we can use genetic techniques that will allow us to compare genetic sequences from different organisms to compare similarities and potential ancestry. So these techniques will have basically will showcase an evolutionary process that species has had according to its molecular evidence, or yes, according to their genetic codes. So basically in here, these techniques are quite remarkable and will give detailed information and overview to define species, but also it will identify the interrelationship between species over geological time. So by using fossil fuels, using the carbon-14, and also the other isotopes that are quite remarkable to identify the evolutionary process that every single species has had. So here, this is an approach and methodology that we have in biology to compare the different morphological species concept against the biological species concept, ecological species concept, and the evolutionary species concept. Okay, so here what we have next is species exist in localized populations, but in some cases they migrate due to other factors, so it's difficult to define it. There is another limitation here, is that species can migrate. This migration can consist of different geographical locations. For example, here we have snail migration, and this is an open population of snails in flower bed. But if they cross this street, the most probable is that a car will crush them. So a close population of snails on traffic island flower bed will be isolated in this case. They will be closed. That will not allow the gene flow and the interaction between their genes and the other snails population's genes. Okay, so roadways can work as a barrier to effective migrations of snails in most cases. But we have also another population of snails that is going to be semi-open population of snails in vegetable patch. When they cross the hedge, which is the home to predators of garden snails, they can face some obstacles in order to reproduce with all the species. So speciation is one of the concepts that will take place here. So speciation comes from local population. So every single local population, as I told you before, will have a gene pool. Speciation and change due to split, mutation that separates the new species from the previous one, establishing every year. So the evidence is the fossil record. So speciation can, okay, we have several examples of speciation. For example, one of them is the northern spotted owl, compared to that one, which is the Mexican spotted owl. So there are like different causes of speciation, but the main ones we're going to mention today are the allopatric speciation and the sympatric speciation. So wolves, for example, that is Canis lupus. In domestic dogs, that is Canis lupus familiaris, are going to represent a certain relationship. One of the examples here is why are wolves and dogs an example of speciation? So the common ancestor of dogs evolved from a wolf-like ancestor about 20,000 to 40,000 years ago through domestication by humans. So genetic and behavioral differences. Over time, dogs develop traits that differentiate them from wolves, such as smaller brains, they increase their socialization with human beings, and reduce fear response. So reproductive barriers, while dogs and wolves can still interbreed and produce fertile offspring, behavioral and ecological differences often prevent natural interbreeding. So as you can see, for example, here, we're going to have like different types of, or subtypes of these species. So for example, we have the Canis lupus lineage. So it was like 53,000 years ago. And there we're going to have other canids such as jackals, does, foxes. The coyote is going to be included here, in the gray wolf. But the gray wolf will have like a different process compared to these other species, because the gray wolf will have a large dog breed, and these are the domestic dogs that we have now, and they are called as Canis lupus familiaris. Also, we have small dog breed. So there is like an interrelationship between these two species. Okay, so the note one, development of barriers within local population is a possible cause. Before separation, individuals, they share a common gene pool, but after isolation, processes such as mutation, and remember that mutations can be three different types: neutral mutation, beneficial or beneficial mutation, and negative. They can cause change in one population, but not in the other. Alternatively, a new population may form from a tiny sample that became separated from a much larger population. So while the number of individuals in the new population may rapidly increase, the gene pool from which they form may have been totally unrepresentative of the original. So with many alleles, there are different expressions of a single gene lost altogether. In this way, populations of the same species can change through accumulation of gradual changes in the genotype through time, so to eventually form new species, genetically distinct species. The second note is going to talk about the smallest unit of evolution is a population. It's really important because normally students misunderstand this concept. So one common misconception about evolution is that individual organisms evolve during their lifetimes, but evolution is not a process that is only taken by individuals. The individual is going to represent the population, but it is the population that evolves in and of itself. Individual organisms cannot evolve, but a population can have genetic variation in traits and can undergo Darwinian evolution. So Charles Darwin, resulting in all the changes in genotypes, determined that were the populations those ones who evolved. Got it? And phenotypes associated with evolutionary changes are going to be closely attached to genotypes. So we never say that I will, okay, this is another common mistake that people say, I'm going to adapt to the Canada's weather, for example. That's not okay. And that's not okay because you don't adapt. Maybe you condition yourself to a certain environment or weather, but the only ones who adapt are going to be the populations. Individuals never go through those processes. Okay, so maybe an individual can go throughout one of those processes, but the interaction between or among the individual with older ones will create certain genetic tendencies, and those genetic tendencies will undergo something that will be called natural selection, and that natural selection can undergo something that is called adaptation and evolution posteriorly. So the concept that is diversity is related to the differences presented in different species. Chromosomes vary in number and shape among organisms. An example can be bacteria. They will have circular chromosomes, whereas animals and plants will have linear chromosomes. And this is closely related to the presence of histones, which are proteins that are located in the DNA. Variety in chromosome number can be related to evolutionary origin and functional differences between organisms. Then we're going to have another concept that is called unity. Unity, the number of chromosomes in the cells of different species varies. But in any one species, the number of chromosomes per cell is normally constant. Then we're going to see a table where we can illustrate this. So here we have the Parascaris quorum, that is the roundworm. The number of chromosomes per cell is going to be 2. We also have the mus musculus, which is a mouse. The number of chromosomes per cell is going to be 40. The Drosophila melanogaster fruit fly, number of chromosomes 8, while the Homo sapiens humans is going to be 46. The Oryzis sativa, which is a rice, 24. Pan troglodytes, which is a chimpanzee, is going to be 48 chromosomes, and so on. So you can observe, for example, the difference between the sunflower, which is 34 chromosomes, and Canis familiaris, the dog, that is 78. So those are like the differences between the unity. Okay, so we have the concept of diversity, we have the concept of unity. Diversity represented by the difference between different species in order of their morphology, of their internal structure, in order of their form. Okay, sorry if I'm being redundant. And unity in number of the differences that exist in the chromosome number, but at the same time the constant information that is in their cells per se. Okay, so here we're going to talk about different structures that are in the DNA. There are the chromatin, that is represented by all the DNA proteins in nucleus. and it condenses to form chromosomes during mitosis. Okay, this is a nucleus and this is a nuclear envelope. So the chromatid, we're going to have sister chromatids, two sister chromatids in every single chromosome, and we'll have the chromosome that is condensed. The chromatid, either two sister chromatids or one chromatid, can be presented here. And okay, so this region is going to be called the gene. So the gene is the unit for inheritance. Okay, so these are like the chromosome terminologies. So chromosomes of a cell occur in pairs called homologous pairs. One of each pair came originally from the male parent and the other one comes from the female parent. So cells in which the chromosomes are in homologous pairs are described as having a diploid nucleus. And this diploid nucleus can be represented by 2n. So 2n means diploid. So where n is going to indicate chromosome. So I multiply this number by the chromosome equals diploid. And we have another type that is one chromosome of each pair, and that will have a haploid nucleus. The haploid is going to be represented by just a singular n, haploid. Okay, so a sex cell has haploid nucleus formed as a result of the nuclear division known as meiosis. Genes and alleles. So locus. Locus is the specific position of a gene. In plural, we say loci, and each gene has two or more forms called alleles. As I had told you, for example, we have the gene that will code the eye color, but the traits, in this case brown eye color, can be expressed by the alleles. So I can have an allele that is going to be related to the brown eye color, but at the same time I would have another different allele of the same gene that will belong to the blue eye color, and I will have another allele of the same gene that is a variation of it that will represent the, I don't know, okay, we have a brown, blue, and green eye color. So the same will happen with the hair color gene. So this band that you observe here is going to be the gene. So the gene, this is the location of the gene, sorry. So this band pattern is going to represent the hair color, and the hair color will have different alleles, different expressions of that gene. So a diploid organism that has the same allele of a gene at the gene's locus in both copies of the homologous chromosomes in its cells is described as homozygous. So similar genes are going to be called homozygous. But a diploid organisms that will have, for example, different alleles, for example, capital letter, lowercase letter, or capital letter, lowercase letter, according to the alleles, is going to be heterozygous. And some of these genes can be expressed, and some of them can be masked. And those genes involve not only physical traits, phenotypes, but at the same time, they involve health condition, health tendencies, and so on. Okay, so let us talk about the next. Okay, so chromosomes are copied identically between nuclear divisions, while the chromosomes are encoded in the form of chromatin. This structure that I previously showed you and cannot be seen. Each chromosome is copied. It is said to replicate. Replication occurs in the cell cycle during the interphase. The two identical structures formed are called chromatids. The chromatids remain attached by their centromeres until they are separated during nuclear division. After division of the centromeres, the chromatids are recognized as chromosomes again. Of course, when chromosomes are copied, the critical event is the copying of the DNA. Double helix that runs the length of the chromosome. Replication occurs in a very precise way, brought about by specific enzymes. So here you can observe, for example, the sister chromatids. They are attached to the centromere, making up one chromosome. And the centromere, a small constriction on the chromatids, is not a gene and does not code for a protein as genes do. Each chromatid is copy of the other with its lineage of genes. Individual genes are too small to be seen, correct? So the centromere is going to divide. The chromatid is separated during nuclear division. And this is one chromosome expressed as if it were two chromatids. We have the karyotyping in karyograms. Human chromosomes of a male, this is the karyotype, and it is seen at the equator of the spindle during nuclear division. So it is then organized in pairs of chromosomes in descending order of size. So we humans will have 23 pairs of chromosomes, where the last pair of chromosomes is going to be the sex pair of chromosomes. In this case, it's a male because it will contain in the pair 23 an X and Y. So X and Y, X and Y will represent the sex chromosomes. But if it was, for example, a female, an individual that belongs, a karyotype that belongs to a female, it would be XX in the 23rd pair of chromosomes. So one of these chromosomes is going to be donated or given by the parent, by the male parent. The other one is going to be given by the female parent. Okay. So these are images of chromosomes cut from a copy of these photomicrographs, can be arranged and pasted to produce a karyogram. Okay, so here we're going to talk about three features that can be used to identify chromosomes. And they are going to be, first, the size of the chromosome, then the bonding pattern, which is the second, how many bonds it has. For example, what is the length of them, and how many of them we can encounter in one region, and so on. And the other one is going to be the centromere position, which is this central structure you can observe here. Human Homo sapiens and chimpanzee, okay. This is a comparison between human chromosomes and chimpanzee chromosomes. So, however, every other living species in the family Hominidae, including our closest relatives, the bonobo, Pan paniscus and chimpanzee pan troglodytes have 24 chromosomes pairs. DNA sequencing has shown that less than 3% of human DNA is different from the chimpanzee. So what might have happened to change the number of chromosomes in humans? Which compare the karyograms of chimpanzees and humans, what do you notice? So an alignment of the human and chimpanzee chromosomes shows that the centromere of human chromosome 2 lines up almost exactly with that of chimpanzee chromosome 2A. So the hypothesis has been suggested that two of the A chromosomes must have fused to form one large one. Let's evaluate the evidence. and the inquiry. So concluding and evaluating. So scientists compare these two chromosomes and found similarities, bonding patterns between human chromosomes 2 and chimp chromosomes that were originally numbered 12 and 13. Human chromosome 2 has two sets of centromeres. Usually a chromosome has just one centromere, but in the human chromosome 2 there are remnants of a second centromere, as you can observe here. So there is a segment of a long arm of human chromosome 2 close to its centromere that has significant similarity to chimpanzee telomeric DNA. The detailed sequence in this region is exactly what you'd expect it to see if the two chimpanzee chromosomes had fused end to end. The evidence supports the hypothesis of an ancestral fusion event between the chromosomes originally numbered 12 and 13 chimpanzees. So this new chromosome is, in fact, the second largest chromosome as viewed under a microscope and is therefore named chromosome 2. The chimpanzee chromosomes involved in the fusion, 12 and 13, have been renamed as 2A and 2B in recognition of this fusion event. So the exact mechanism through which these two chimpanzee chromosomes fuse is not yet completely understood, although studies show that no genes from the ends of chimpanzee chromosomes 2A and 2B appear to have been lost. So there you will observe, for example, the centromere that is in the middle. And it just perfectly fits with the human chromosome 2. So chimpanzee chromosomes 2A and 2B will be closely related or equal to human chromosome 2. So this is an evidence for evolution of human chromosome 2. Okay, unity and diversity of genomes within species. The genome is all the genetic information of an organism. Organisms in the same species share most of their genome, but variation occurs which gives some diversity. In humans, the most common genetic variation is called single nucleotide polymorphism, and it can represent a difference in a single nucleotide. For example, you can replace in the chain of nucleotides in the order of nucleotides one you can replace it by another nitrogenous base that is adenine, for example. And this can occur throughout the genome. On average, approximately once in every 1,000 nucleotides in humans, which means there may be between 4 and 5 million single nucleotide polymorphism in the genome of one person. So this single nucleotide polymorphism may be unique or occurs in many individuals. And scientists have found more than 100 million single nucleotides polymorphism in populations around the world. These variations are usually found in the non-coding parts of DNA found between genes. In this way, single nucleotide polymorphisms contribute to human diversity. So, single nucleotide polymorphism occurs within a gene or in a regulatory region near a gene. They may play a more direct role in disease by affecting the gene's function. Scientists have shown that certain single nucleotide polymorphism may help to predict an individual response to specific drugs and risk of developing particular diseases. Single nucleotide polymorphisms can also be used to trace the inheritance of genetic diseases within families. Genomes vary in overall size, which is determined by the total amount of DNA. Genomes also vary in base sequence. Variation between species is much larger than variation within species, so you can have different variations inside the species, but also between species. And genetic variation plays a key role in allowing us overwhelm and face the different obstacles and environmental pressures in order to evolve. Common mistake: DNA in chromosomes and in mitochondria are considered part of the genome. as well as chloroplast DNA in plants, but not messenger RNA and transfer RNA. And I would like to talk about the Human Genome Project. The Human Genome Project was undertaken um, by scientists to map the entire human genome. It was a publicly funded project that was launched in 1990. The ultimate objective of the Human Genome Project was to discover the base sequence of the entire human genome. The work was shared among more than 200 laboratories around the world, avoiding duplication of effort. On 26 June 2000, an announcement established that the sequencing of the human genome had been achieved. At the same time, the Human Genome Project was underway. Teams of scientists set about the sequencing of the DNA of other organisms. Initially, this included the common human gut bacterium that is known as E. coli, the fruit fly, the mouse, baker yeast, and Arabidopsis wheat related to important crops such as broccoli and oilseed rape. Since then, scientists have sequenced the genome of about 3,500 species of complex life with around 100 being sequenced at reference quality, which is used for in-depth research. Here, the order of the nucleotide bases is being determined in a molecule of DNA by a largely automated process. Nowadays, with artificial intelligence, all of these data are being inserted in a great platform where all the scientists are able to access this information. Okay, genes, amount, and function. Genes may be located in base sequence data, but the detection of sequences that are uninterrupted by start and stop codons. Such regions are more likely to code for a protein. The 3 times 109 bases that make up the human genome represent about 20,000 genes, far fewer than was expected. Drosophila, the fruit fly, has almost half or number of genes, and a rice plant has more than 40,000 genes. Promoters and enhancers are associated with many genes and may determine which cells express which genes, when they are expressed, and at what level. The genetic code of DNA is not altered by the environment, except in cases of mutations. Information flows out of genes and not back into them. However, while our genes are largely immune to direct outside influence, our experience may regulate the expression of particular genes. For example, genes are switched on and off by promoters, and these may occur in response to external factors, environmental pressures, for example. If so, our genes may be responding to our actions as well as causing them. Okay, so also genetic disorders location. Another outcome of this Human Genome Project is the ability to locate genes that can cause genetic disorders in humans. Okay, some of them can be expressed, but other ones are going to be masked. And normally, more than half of all genetic disorders are due to a mutation of a single gene that is commonly recessive. I mean, it's not expressed at all. So to prove a gene is associated with a disease, it must be shown that patients have a mutation in that gene, but that unaffected individuals do not. The outcome of the mutation in people is a loss of the ability to form the normal product of the gene. Common genetic disease included cystic fibrosis, sickle cell disease, and hemophilia. So genetic disorders location is one of the greatest outcomes that this project has given us. I would like to thank you. I want to give you congratulations if you have reached, if you have understood all the lessons. I know that these cards can be charged of many content, but believe me, it works. Every single thing works here. I hope you have understood many things about diversity, and we're going to continue with our project in order to educate more students in biology and science. See you.